lecture we will see the biosynthesis of aromatic amino acids, tyrosine, tryptophan and phenylalanine. Glycolytic intermediates PEP, phosphoenol pyruvate and erythrose 4-phosphate condense to form 3-keto-3-deoxy-arabinoheptylosonate 7-phosphate, also known as DAHP, which cyclizes to form corismate, which acts as the precursor for the synthesis of aromatic amino acids. In this pathway, as this pathway occurs only in plants and microorganisms, it acts as a natural target for herbicides. The final two reactions of tryptophan biosynthesis are catalyzed by tryptophan synthase. The alpha subunit consisting of 268 amino acids of this alpha 2 beta 2 bifunctional enzyme cleaves indole 3 glycerol phosphate yielding indole and glyceraldehyde 3 phosphate. The beta subunit consisting of 396 amino acids joins indole with L-serine in a PLP dependent reaction to form L-tryptophan. X-ray structures of tryptophan synthesis from Salmonella typhimurium shows that the protein forms a 150 angstrom long two-fold symmetric alpha-beta-beta-alpha complex in which the active site of neighboring alpha and beta subunits are separated by 25 angstroms. These active sites are joined by a solvent-filled tunnel that is wide enough to permit the passage of the intermediate substrate indole. Indole is only released when the beta subunit is ready to accept it. Elimination of water from the serine PLP skip base based on the beta subunit to form an amino acrylate PLP skip base intermediate triggers a conformational change that activates the alpha subunit to produce indole. This is the reaction mechanism of the active site. Now let us see the flow of the pathway. Phosphoenol pyruvate as discussed along with erythrose 4-phosphate condenses to form DAHP. DAHP in the presence of dehydroquinolate synthase reduces NAD to NADH and forms 5-dehydroquinolate. This 5-dehydroquinolate releases a molecule of water in the presence of 5-dehydroquinolate dehydratase, forms 5-dehydroshikimate. This 5-dehydroshikimate oxidizes NAD and in the presence of shikimate dehydrogenase forms shikimate. This shikimate in the presence of shikimate kinase forms shikimate 5-phosphate. Here a molecule of ATP is converted to ADP and the phosphate is attached to the O. Shikimate 5-phosphate in the presence of EPSP synthase and from shikimate 5-phosphate a lone pair of electrons attacks the carbon of the phosphoenol pyruvate, thereby releasing a molecule of inorganic phosphate. 5-enol pyruvyl shikimate 3-phosphate in the presence of shikimate synthase releases inorganic phosphate and forms corismate. Corismate is the precursor for all the 3 aromatic amino acids. Firstly, we will discuss the biosynthesis of tryptophan from corismate. Corismate in the presence of anthranilate synthase forms anthranilate. In this step, glutamine is deaminated to glutamate and a molecule of pyruvate is released out. Anthranilate in the presence of anthranilate phosphoribosyl transferase takes in PRPP and releases PPI, thereby forming N5' phosphoribosyl anthranilate. N5' phosphoribosyl anthranilate in the presence of anthranilate isomerase forms enol 1O carboxyphenyl amino 1-deoxyribulose phosphate, which in the presence of indole 3 glycerol phosphate synthase forms indole 3 glycerol phosphate. Here a molecule of water and carbon dioxide are released out. Indole 3 glycerol phosphate in the presence of tryptophan synthase alpha subunit releases glyceraldehyde 3 phosphate and indole group. This indole group in the presence of tryptophan synthase beta subunit takes in a molecule of serine and releases water, and tryptophan is formed. This chorismate in the presence of chorismate mutase forms priphenate. Priphenate in the presence of priphenate dehydrogenase, NAD gets reduced to NADH and a molecule of carbon dioxide is released out, thereby forming 4-hydroxyphenyl pyruvate. This 4-hydroxyphenyl pyruvate in the presence of aminotransferase glutamate gets converted to alpha ketoglutarate and thereby forming tyrosine amino acid. And this priphenate, if it takes another path like priphenate dehydrogenase acting on priphenate giving rise to phenyl pyruvate, a molecule of carbon dioxide and OH minus are released out and phenyl pyruvate is formed. Phenyl pyruvate in the presence of aminotransferase glutamate gets converted to alpha ketoglutarate and phenylalanine is formed. Phenylalanine amino acid is formed. Thank you.